Hello students and happy Thursday. Go ahead and pause your screen to think and answer the spell work. So today we are going to think about the pieces of communication. By communication I mean cell to cell communication to answer the following questions. We're talking about ligands and target cell receptors. So how could the fluctuation and the concentration of each of these, talking about ligands and target cell receptors, change the message strength, duration, and ability of a target cell to receive the message? So think about this and write a few sentences. Basically, it's asking what would happen if there's more ligand or less ligand? How would that vary based on the message or the strength or the duration or the target cell's ability to receive the message? And how would the concentration of target cell receptors on the plasma membrane of the target cell also influence these variables? Go ahead and pause your screen to answer this. I next wanted to talk about the body mechanisms assignment. It used to be called the cell communication assignment, but many students were confused. This cell communication or body mechanisms assignment is not the same as the discussion board. The discussion board will be independent work where you have an initial reply post and a subsequent peer reply post. Right here is the instructions. There are your assigned topics, whereas our body mechanisms assignment has not had thorough instructions yet, but that will be a group assignment where you can sign up with people that you wish to. Right here in the body mechanisms assignment, you can view here, click on which one you want to join, but if you don't join by Friday, one will be selected for you. All right, so we have talked about the different ways that cells can communicate with each other, but what happens after that target cell receives the message? So the ligand will either bind to a receptor on the target cell's plasma membrane, or if it's able to, it will go through the plasma membrane and bind to a receptor that's in the intracellular environment. So events will occur after the binding of the ligand to the receptor. After the cell receives that response that a ligand has bound to the receptor, it creates a subsequent change within the target cell in response to this signal. This is called signal transduction. Different cell types can respond differently to the same exact signal. So we're going to go through this a little bit because this can be a difficult topic to understand. Although epinephrine can be a ligand with a specific receptor, not all cell types have the same response or signal transduction to that ligand receptor bonding that occurs, or complex sometimes it's called. The ligand receptor complex can cause different cell responses based on the specific type of cell that the target cell is and what properties make that cell have a specific response. So for example, epinephrine, this is considered the fight or flight hormone, and this is a catecholamine released from your adrenal glands. These adrenal glands are located directly superior to the kidneys, and they have many effects on the body. For example, when epinephrine binds to the beta adrenergic receptors of the heart, it's going to increase our heart rate. When epinephrine is binding to the target cell of our muscles, for example, it's going to have a different response. It's not going to increase our heart rate. It's a muscle cell. Muscle cells can't have that type of effect directly on the beta adrenergic receptors of the heart. Instead, what happens with those cells, our muscle cells, is they're able to take stored glycogen, which has a storage of energy in our muscles, and it's going to be able to break down that glycogen to give the muscles an extra energy source. Epinephrine also acts on muscle cells by decreasing the amount of glucose that is going to be synthesized into glycogen. So it has two mechanisms that are going to increase our cellular availability of glucose, which in turn gives our muscles increased energy. Although it still follows this fight or flight pattern where now we're worried about having enough energy to supply our muscles with sufficient fuel to take on their functions, we can see that the heart racing and having increased energy for our muscles are two completely different functions that two different cells responded differentially to the same epinephrine signal. Glucagon has a similar ability to have different responses of the target cell in response to the ligand receptor complex being formed. So glucagon is a hormone as well, and glucagon is secreted by the pancreas. Glucagon is secreted by the pancreas when our blood sugar is getting too low. And glucagon can act in multiple ways to increase the supply of glucose to the bloodstream. Glucagon can act on the liver, for example, to have the target cells take their glycogen storage of glucose, which is stored as glycogen in the liver, and it's going to ask the liver to break down that glycogen so that we can have more sugar within our blood. On the other hand, glucagon can also act on other cells to have gluconeogenesis occur, which is creating glucose from alternative fuel sources, such as amino acids, which we would do in times where we are very low on blood sugar and we are desperate for an energy source that our body can use. And here is again that chemical signal that we just talked about, epinephrine. It's a hormone which would have the endocrine type of communication across the body. Epinephrine is considered the fight or flight hormone, and it has differing effects depending on the target cell. But different sets of proteins respond to that signal depending on the target cell. For example, the liver mobilizes glucose. It's able to perform glycolysis, and break down the storage form of glucose, which is glycogen, within our liver and take it into its original monomers to form glucose monomers, which our body is more easily and readily available to use. Epinephrine at the same time causes the heart to contract more forcefully and at a faster rate of speed. And it also can cause our blood vessels to not only contract, but it also can cause our blood vessels to dilate. And here is the hormone glucagon, which is another chemical signal, and it is from the endocrine type of cell signaling. A variety of cells respond the same way to control blood glucose. We're going to increase the blood sugar when we release glucagon. Glucagon has a target cell of liver cells and also a target cell of adipose tissue. So adipose tissue is another place where we can store fat, and fat is a source of alternative fuel when we are out of glucose. So fat can also undergo gluconeogenesis, and therefore on the liver it's going to act on our glycogen storage, yet on the adipose tissue it's going to act on our triglyceride storage, which is fat storage within our adipose tissue. And this performs either gluconeogenesis in the adipose tissue, or it can perform glycolysis, acting on the glycogen storage in the liver cells. Phosphorylation is a signal transduction pathway to which, after the ligand receptor complex is bound on the target cell, the addition of a phosphate group changes the target cell's response. So this phosphorylation is going to inactivate or activate a protein frequently through this process of phosphorylation. Phosphorylation changes the activity of a protein via multiple ways. So protein kinase, and remember this ASE always is going to tell you that it's an enzyme. So protein kinase is an enzyme that adds a phosphate to a protein. Phosphatase is an enzyme that removes a phosphate from a protein. So these two mechanisms of phosphorylation, which is either adding or subtracting a phosphate group from a protein, are going to completely change the protein's function and therefore will activate this signal transduction pathway to elicit a certain response in a target cell. So here is an example when we have ATP and then you add kinase, we undergo phosphorylation and it becomes from adenine triphosphate, we lose a phosphate group and it becomes adenine diphosphate or 2 phosphates. And then we can see that it changes the chemical structure, and remember that structure follows function. So because we have a different structure, it can be thought of as a different function occurring. Now, if we add the enzyme phosphatase, we are going to add a phosphate group off of that, we're going to lose the phosphate group and change the structure. So this is one easy way to show, okay, well, if we want to signal a cell, how do we stop that signal from happening? Well, in one direction, we can just add the enzyme kinase, and that's going to change ATP to ADP, and it's going to phosphorylate or add a phosphate group to this structure. On the other hand, if we simply add the enzyme phosphatase, we can lose that phosphate group and regenerate this protein or amino acid back into its original form. So instead of having that signal alter that amino acid indefinitely, there's a process where the amino acid is able to go back to its original form. It's a forward and backward reaction to where enzymes can act in a forward or backward way to undergo different functions to carry out the chemical messages that are sent to target cells. Okay, next we're going to talk about receptors. Receptors can be defined by their location. They can be intracellular receptors. Intra means within, so intracellular receptors are within or inside of the cell. They're within the intracellular fluid or intracellular space. You can also have cell surface receptors or a membrane receptor, which are embedded on top of the plasma membrane to bind to a ligand which is present outside of the cell. Transmembrane protein in contact with both the cytoplasm and the extracellular environment. So you can have a transmembrane, trans means across, so you can have proteins that are both on the outside and the inside of the environment to where they're able to communicate with structures inside the cell and bind to those on the outside of the cell. Intracellular receptors, one example of these are hormones. Steroids are common nonpolar lipid structures, and they are embedded throughout the cell or plasma membrane. They can freely move across the plasma membrane and attach to a steroid receptor that is within the internal environment or the intracellular fluid or the intracellular space of the target cell where there is a receptor present. The binding of a hormone to the receptor causes the complex to shift the cytoplasm to the nucleus. So basically, we have this receptor ligand complex, like I was talking about, where the ligand is the hormone that binds to an intracellular receptor and makes the cytoplasm, the jelly-like substance within the cell shift towards the nucleus. This acts as a regulator of gene expression, so it can help turn on genes or turn off genes, which we'll get more into that when we talk more about genetics and gene expression. Here's an example of our intracellular receptors. So you have these hormones, and since they have the hydrophobic nature, similar to that of the hydrophobic tails, it is able to move freely through the plasma membrane. And these hormones can go directly through the plasma membrane without a transmembrane protein or channel. And while they enter the cell, they find their ligand-specific intracellular receptor, and they form a complex with this intracellular receptor. And therefore, it changes the complex where a site is now exposed that was once blocked, which will be able to change the gene expression. The main point of this, although this is a more difficult slide than it seems, is that hormones can bind to intracellular receptors and have the ability to change intracellular complexes that will make binding more difficult or more easy, depending on where the hormone binds to. Okay, there are 3 functional domains of a steroid receptor. So if we go back to this picture, it's going to explain a little bit better. So we have the hormone binding domain. So you can see that there's this intracellular receptor here, and there's a specific part of this receptor protein that has that lock and key fit to the hormone, and that is the hormone binding domain. Next is the DNA binding domain. So on the second part of this receptor protein, there is a DNA binding site, but before the hormone bound to this receptor, that DNA binding site was inhibited. There was nothing available to be bound to because it was blocked by an inhibitor. And the third part is the domain that interacts with coactivators to affect the level of gene expression. In its inactive state, the receptor typically cannot bind to DNA. This receptor complex, however, after activated, frees up the inhibited site where the protein because an inhibitor protein occupies the DNA binding site, is now in a different conformation of this complex. Binding of a ligand changes the conformation of this complex and therefore has an increased ability to bind to that DNA binding site. So now, because of this hormone coming into the cell, it caused a reaction to happen where basically it unlocked the complex, and the inhibitor is no longer blocking the DNA binding site and has an easier ability to go and to the site into the nucleus and have a greater influence on gene expression. And that's where we're going to leave off our lecture today. Stay tuned for your exit ticket. All right, everyone, here's your exit ticket. I hope you all have a wonderful day, and I look forward to seeing you all next week.